My name is Josh Perfetto, and I'm a biohacker and biotech entrepreneur. A biohacker is somebody who does something really new and novel with biology. Just like you might kind of hack together in an electronics project or a software project, we're starting to be able to do the same thing with biotech. Biotech is the application of technology to biology. I'm really an engineer at heart, which means I like to build things. What really sparked my interest in biotech is when I found out that we could engineer biology in the same way that we engineer software and electronics. I've started a couple biotech companies. One is OpenPCR, which is to make a DIY thermocycler that you can build yourself. Another biotech company I've started is Cofactor Bio, which is making kits and a DNA barcoding service. DIY means do it yourself, and a really new thing is DIY bio, in the concept that you can actually hack a microbe yourself in your home. OpenPCR is a thermocycler, which is a machine that you can use to replicate DNA. PCR stands for the polymerase chain reaction. PCR is a really common lab technique that is used for everything from doing medical diagnostics to doing genetic engineering. The commercial thermocyclers that would do PCR can cost $3,000, $5,000, and upwards from there. And that puts it out of reach of a lot of schools, a lot of hobbyists, and even small biotech companies. So OpenPCR was a project to really reduce the price of a thermocycler and make it much more widely available. We came up with the idea of a PCR machine you could build yourself. Part of OpenPCR is to make an open machine. So that means the design is on the internet. You can get all the details of how it works. So you can actually hack the OpenPCR to make it do something different. But you also can buy a kit, and it has everything you need to build your own PCR machine. I have a small lab in my house where I developed the OpenPCR machine, did a lot of the design and the testing. But when it comes to actually assembling the OpenPCR machines, we do that here in the garage. The OpenPCR kit costs $599, and it has everything you need, including the instructions, the case, the components, to build your own working PCR machine. And what the PCR machine basically does is it has to very accurately control the temperature of a block. The core part of OpenPCR is a device called a Peltier, which is a solid-state heat pump. So that means you run current one way through it, and it will pump heat up. You run it the other way, heat goes down. So we use this to heat and cool this block where you put your PCR reactions in. The core Peltier is controlled by an Arduino microcontroller. And another important thing we have is a heated lid, which is necessary to prevent condensation from occurring on the tubes. You could read your DNA. You could use a commercial service like 23andMe to do that, or you could actually genotype yourself in your house using PCR. You could detect whether the tomatoes you're buying are GMO tomatoes or not. A GMO is a genetically modified organism. The term commonly comes up when we talk about food because genes were added to certain types of food to give them desirable traits so that it may grow better or it may be more resistant to pests. You can do DNA barcoding and detect what species of fish did you buy. DNA barcoding is a way to identify the species of an organism from a tissue sample. It came about when we wanted to make it really cheap and easy to identify what species something was. So a taxonomist who specializes in turtles has identified this type of turtle, and then they put it in a database. So now we have a public database that has all the barcodes that basically anybody can go and read against. So I find a turtle and I want to know what it is, and I'm not an expert in turtles. I can use barcoding to sequence that gene, look it up in the database, and then identify what that turtle is. We'll use a piece of salmon, and we'll extract DNA from the salmon, amplify it, and then sequence that, and then identify exactly which type of salmon it is. I'm going to get my extraction buffer and put 70 microliters of that into this PCR tube. We're now going to isolate out a very small piece of this fish. We'll put this into the extraction buffer, and I'll just mash it around a little bit like that. We have the PCR reaction that we've prepared, and now it's ready to go inside the open PCR machine to start the thermal cycling. So I'll put it in the machine there, close the lid, and create a new experiment. All right, we're gonna define the PCR reaction. The first step is denaturing, which is where the two strands of DNA will come apart. So I'm gonna set it to 95 degrees Celsius for 30 seconds. The next step is annealing, and that's where one of the primers will go find their match on the single strand of DNA and kind of anneal the region of DNA that we want to amplify. And the third step of PCR is extension. That's where the polymerase will kind of latch onto that primer, and it will start extending down the template, actually copying the DNA, which is what doubles the amount of DNA each time. And I'm going to define that we're going to do 30 cycles. So it's going to denature, anneal, and then extend 30 times. Every time it does that, it will double the amount of DNA. So theoretically, we could get 2 to the 30th more DNA than we put in. And then we'll sequence that to read the barcode region. We'll then look that up in a database where barcodes of like known organisms have already been populated, and we'll look for a match. And once we find that match, we know what species of organism we started with. We can learn from DNA barcoding how widespread certain animals are. We can gather data from like kind of across the country and actually start to plot this on a map. Where do different species live? The limiting factor is actually not how much we can read the DNA, but how much we know what it means. And what I ultimately hope is that by reading the DNA from the world that's all around us, we can learn a lot about our world that way and make better decisions.